ribonucleic acid, that is RNA. RNA molecule is also composed of nucleotides like that of DNA. There are few differences that we should note between RNA and DNA. That is, the sugar component of RNA, as we know that nucleotides are composed of phosphate, sugar, and base. So the difference between RNA and DNA is that the sugar component of RNA is ribose, whereas in case of DNA it is deoxyribose, that means lacking oxygen. So this deoxyribose is a component of DNA and ribose is a component of RNA nucleotide. Second difference, instead of pyrimidine thymine, so this thymine is there in case of DNA. So instead of this thymine, RNA contains pyrimidine called as uracil. So there is absence of thymine in RNA. That is the second difference. Third, in most of the cases we find that DNA is double stranded, whereas frequently RNA is single stranded. Then there are three types of RNAs. They are called as tRNA, that is transfer RNA, rRNA, that is ribosomal RNA, and mRNA, that is messenger RNA. RNA and DNA both are present in all biological cells. The exception is virus. Virus is not a cell. Virus contains either DNA or RNA. Now we'll see the functions of both DNA and RNA. Briefly, mRNA. These are synthesized by enzymes using information code in DNA, and this is the first step in the protein synthesis. Second is tRNA. tRNA are the molecules that are quite stable and has a typical clover leaf structure shape, and these tRNAs are responsible for carrying out amino acids, and these amino acids are added to growing polypeptide chain. rRNA. So about 80% of total cellular RNA, rRNA comprises 80%. These are associated with ribosomes and therefore they are called as R. And ribosome, it is a site for synthesis of protein. Now in detail we learn about the functions of DNA, mRNA, rRNA and tRNA. This is a bacterial cell, a prokaryotic cell, as we know that this is single circular covalently closed DNA which is double stranded. There is formation of a fork. One of the strands of this DNA is copied and a complementary strand is synthesized by an enzyme called as RNA polymerase. This RNA polymerase enzyme catalyzes the formation of mRNA. So mRNA is formed because of RNA polymerase. This mRNA is formed from this one of the strands of double stranded DNA by RNA polymerase. So the product of RNA polymerase reaction is mRNA. Then this mRNA binds to ribosomes. This is ribosome, and then amino acids are added through tRNA. So at the bottom you can see these are amino acids. So amino acids, so as this mRNA, this is the mRNA, so this ribosome, it moves from this direction to this direction. That means this is 5 prime end of mRNA. So it is moving from 5 prime to 3 prime. So this ribosome, it moves, and as it moves, it leads to synthesis of a polypeptide chain. So it moves on. So the polypeptide chain, it grows due to the addition of amino acids. It moves on and the polypeptide chain is growing. This is how a polypeptide chain is synthesized. So briefly it is shown in this diagram. This is the process, whole process of protein synthesis, which involves formation of mRNA, that is called as transcription, and formation of this peptide chain, it is called as translation. So this is in case of bacterial cell. In case of eukaryotic cell, similar events are there. Only the difference is that in case of eukaryotic cell, there is a compartment. This compartment is called as nucleus, and this nucleus contains double-stranded DNA with free ends. So similar to the earlier process, what we have seen in case of this bacterial cell, the first step is transcription. So as a result of transcription, RNA polymerase binds to this DNA, and one of the DNA strands is used as a template, and that leads to formation of mRNA. So mRNA is formed. Then this mRNA, it enters into cytoplasm through pores which are present in this nuclear membrane. After reaching into cytoplasm, this mRNA, it binds to ribosome. Then these ribosomes moves ahead, and that leads to synthesis of polypeptide chain, that is from 5 prime to 3 prime. This is the detailed diagram that shows protein synthesis. You can see this is the mRNA, and this mRNA has these codons. So we have already learned that the codons are triplet. That means each codon is made up with 3 nitrogenous bases, and the nitrogenous bases that binds to these codons will be called as anticodons. We can see that this mRNA, it binds to this ribosome. Then in cell we have tRNAs. One thing is there that this ribosome, it also contains RNA, and that RNA helps in the binding of this mRNA to ribosome. So I'll repeat, the RNA which is present in ribosome, it helps in the binding of mRNA to ribosomes. So the RNA which is present in ribosome, it is called as rRNA. Function of rRNA is it helps in the binding of mRNA to ribosomes. Fine. Then, and we also know that this mRNA, it carries the information, it carries the information which is there in this DNA. So that is the function of mRNA, and that is why it is called as a messenger RNA. It carries the message from DNA. In cell we have thousands of tRNAs, and each tRNA, this is the clover leaf structure, this is a nucleotide chain, this is all nucleotide chain, and these three are just highlighted. These three are nitrogenous bases. So nitrogenous bases are present everywhere, here, everywhere. Only these three are highlighted because they are responsible for pairing with this codon which is present on mRNA. That's why they are highlighted. So everywhere this is nucleotide chain, and this nucleotide chain, it has nitrogenous bases. Only these three are shown here. So each tRNA, is bound to a specific type of amino acids. So there are thousands of different types of RNAs, and each tRNA is carrying specific amino acid, and it is also carrying a specific anticodon which is complementary to the codons or the nitrogenous bases which are present on mRNA. That is why they are called as anticodons. Codons are present on mRNA, anticodons are present on tRNA. So whatever is the sequence of codon present here, the complementary tRNA, which is having this complementary nitrogenous bases anticodon structure, will come and it will bind here. And each mRNA, it carries one amino acid. Now here in this diagram, what is shown that this is growing polypeptide chain. This mRNA has already bound here. It is carrying amino acids, a peptide chain, which are bound together by peptide bonds. Now this mRNA, this tRNA carrying anticodons complementary to this codon will appear here and it will bind here. Then this ribosome, it will move ahead in this direction, leaving this tRNA behind. So what will happen that this tRNA will come at this position because this is moving, and this whole polypeptide chain is transferred to this amino acid, so that this growing polypeptide chain will be transferred to this amino acid through formation of a peptide bond, and this reaction continues. So this ribosome, it moves ahead, tRNAs are continuously adding in, and there will be continuous synthesis, transferring of this growing polypeptide chain to new amino acid through peptide bond. Now we'll see this through an animation which we have already learned in the scope of microbiology in the last semester.
Is from DNA to protein. So this event happens in two stages. First is transcription, in which DNA to mRNA synthesis is the step. Then the second step is RNA to protein. That means from mRNA to protein. That is called as translation. Now let us understand the process of transcription and translation. So this process involves the role of three types of RNAs: mRNA, that is messenger RNA; rRNA, that is ribosomal RNA; and tRNA, that is transfer RNA. mRNA is synthesized from the DNA, which is called as messenger RNA to protein. Suppose this is a cell, that is eukaryotic cell. It contains a nucleus, and this nucleus, it contains chromosomes. This is called a genome. This genome, it consists of chromosomes. These chromosomes are made up with long strands of DNA which are wound around the histone proteins. This DNA, it contains genes. These genes contains the instructions responsible for the synthesis of protein. So there is an enzyme called RNA polymerase. It binds to that gene and starts synthesis of messenger RNA. Here you can see that one of the bottom strand of that double-stranded DNA, it is so after unfolding that bottom strand, is being used as template for the synthesis of new strand. So the new strand which is being synthesized here, it is the mRNA strand which is being synthesized by RNA polymerase. So you can see here the nitrogenous bases which are being added are adenine, uracil, guanine and cytosine. So these nitrogenous bases are added for the synthesis of mRNA. So this is the synthesis of mRNA. This is transcription. In case of eukaryotic cell, this messenger RNA may contain unnecessary bases, nitrogenous bases, which are called as introns. They are cut by nucleases, and this is how there is synthesis of then this mRNA, it comes out of nucleus in cytoplasm. That happens in case of eukaryotic cell. So these are the ribosomes that binds to messenger RNA, and this messenger RNA, then there is presence of tRNA. You can see the structure of tRNA, transfer RNA. At the top there is presence of amino acid, and at the bottom you can see there are sticks, color sticks, green, white and orange. These color sticks are nothing but the nitrogenous bases. So there is presence of codons in DNA, and these are the complementary bases which are called as anticodons. So whatever the codons which are present, which are transferred into mRNA, these are the anticodons. That means these anticodons are going to bind to the specific codons. This is called as genetic code. This genetic code is triplet, and we know that there are 20 different types of amino acids. So this sequence of anticodon, it determines the addition of amino acids. So this tRNA then binds to the specific codons present in mRNA, and then it goes on adding these. They're responsible for the delivering of amino acids, as you can see in this picture, that first tRNA, it is getting bound and it is then transferring that amino acid to the growing polypeptide chain. This is how it happens. This is growing polypeptide chain that you can see. This is how. So once the last amino acid is added, then it leads to formation of a particular shape of that polypeptide chain, which is called as protein.